Somewhere in the Middle of America. You know that song, right? Okay, well, I'm going to sing on the mic. It's Counting Crows. Omaha, somewhere in the middle of America, getting right to the heart of matters. It's the heart that matters more. I think you better turn your ticket in and get your money back at the door. All right, let's move on. I'm going to cut that out. Oh, yes, I am. That's 30 seconds of it. All right, so we finished up with the TCA cycle. So the first couple slides here are just kind of reminding about some of the aspects we finished up with. So remember, we talked about how it's really important for the cell to be able to maintain constant concentrations of all of these TCA cycle intermediates, right? If we don't maintain these concentration levels, then the cycle can't function properly. This is especially true. We talked about how the concentration of oxaloacetate has to be really low for otherwise basically like to be a good control mechanism for the TCA cycle. So we need steady concentrations of all these intermediates, which seems reasonable, right? Because we talked about how in the TCA cycle, 2 carbons come in the form of acetyl-CoA and 2 leave in the form of carbon dioxide. But, right, so for the cycle itself and thinking about catabolism, then that's pretty easy to figure out how that happens, right? But a lot of these intermediates are also the origin, the starting point for pathways that branch off. Oxaloacetate is used to be converted to aspartate, right? So that's how we make aspartic acid, which is then used as the precursor for lots of other amino acids and nucleotides. Same thing goes for alpha-ketoglutarate. It's the precursor for glutamate, which is then used to build other amino acids and purines. Okay, citrate becomes fatty acids and sterols. Succinyl-CoA becomes porphyrins and other molecules. Right, so the idea is that these concentrations get depleted during anabolic times, right? To build up other molecules, we deplete the TCA cycle intermediates, and this can't happen for if catabolism is going to continue, if we're going to be able to process carbon all the way to carbon dioxide. So there are these other reactions called anaplerotic reactions that replenish the TCA cycle. And we talked about 3 main pathways. One is we've already seen in gluconeogenesis, we can convert pyruvate directly to oxaloacetate using pyruvate carboxylase, right? That's an anaplerotic mechanism that consumes an equivalent of ATP or GTP. Okay, so we consume some energy to carboxylate pyruvate to become oxaloacetate. That's a viable pathway to replenish the TCA cycle. In addition, we saw in gluconeogenesis how we convert oxaloacetate to phosphoenol pyruvate. Well, as it turns out, that can be reversed as well using the same enzyme, PEP carboxykinase. The cool thing about PEP carboxykinase, right, is reversing this will actually produce an equivalent of ATP. So we know in gluconeogenesis, decarboxylating oxaloacetate and then phosphorylating the product consumes energy. Well, reversing that produces energy. Okay? There's also another mechanism to convert PEP directly to oxaloacetate called PEP carboxylase. So overall, the net product is the same, right? It's the interconversion of PEP and oxaloacetate. However, this one does not couple energy, right? So there's no energy consumption or production in this step. So it's not as ideal for some organisms. And the last path we talked about was this enzyme called the malic enzyme, which is able to directly convert pyruvate to malate. And this enzyme is present in a lot of different eukaryotes and bacteria. Okay, we won't talk about its mechanism at all, but it is a very important and viable anaplerotic step. Okay, that's kind of where we ended up on Thursday of last week. One other thing that I want to mention that's related to the TCA cycle is in a lot of organisms, especially like some bacteria and plants. This is really important in plants. It's how basically seeds can survive just on the fat that's stored in the seed. These organisms can live only on acetate, right? We're going to see later in the semester that the breakdown of fatty acids results in the production of acetyl-CoA. We break down fatty acids 2 carbons at a time, and we end up with a bunch of equivalents of acetyl-CoA. Well, plants and other organisms are able to use just that fat breakdown product to survive on. okay? And it's a cycle that's very much related to the TCA cycle. And in fact, it uses a lot of the same enzymes, okay? So the idea here is that we take 2 equivalents of acetyl-CoA, so overall 4 carbons, and we pump them through this cycle and produce succinate. Succinate, of course, is a TCA cycle intermediate, so this succinate can then be used as a carbon source that can be funneled back into gluconeogenesis to make sugars from. Okay, so the idea here is it starts off exactly the same as the TCA cycle. We condense oxaloacetate with acetyl-CoA and we make citrate. Nothing surprising here. Citrate is isomerized to isocitrate using the same enzyme as the TCA cycle, aconitase. At this step, though, we break away. So instead of oxidatively decarboxylating isocitrate like we do in the TCA cycle, we use isocitrate lyase and we cleave this into 2 fragments. We start with a 6-carbon fragment, we end up with a 4-carbon fragment, that's our succinate, and we end up with a 2-carbon fragment that is an alpha aldehyde. So this is glyoxylate. And then the glyoxylate really in many ways kind of acts like oxaloacetate at this step. We can activate acetyl-CoA to be a nucleophile, attack the carbonyl, the alpha carbonyl here of glyoxylate, and instead of making citrate, we end up making malate. So we condense together 2 2-carbon units, 1 acetyl-CoA, 1 glyoxylate, and that gives us malate, which can then, we know, be used to regenerate oxaloacetate. Okay, so the cycle is very much related with just 2 different enzymes, isocitrate lyase and malate synthase. Malate synthase is mechanistically related to citrate synthase. Isocitrate lyase is mechanistically related to one of the glycolytic enzymes. Which one, do you remember, y'all remember which is the lyase? Which one splits things in 2? We take fructose 1,6-bisphosphate, split it into, what enzyme is that? Not enolase, it's aldolase, yeah, the aldolase. So this you might imagine is somewhat mechanistically similar to aldolase because they're both lyases. Okay, so the overall reaction here is 2 equivalents of acetyl-CoA, so 4 carbons producing a succinate, a 4-carbon unit. And in the process, we also reduce NAD plus to NADH, so that can be coupled to energy production as well. And this succinate, this 4-carbon unit, can then be used to go on to make other things. It's viable to push this off into gluconeogenesis so we can make sugars or whatever else the plant or other organism needs to make. We cannot do this, right? We do not have these enzymes. Mammals don't have these enzymes, so we cannot live just on fats, because if we break down fats, we can use them to make energy, but we have no path for converting acetyl-CoA back into sugars. Okay, acetyl-CoA cannot go back into gluconeogenesis because both carbons in acetyl-CoA that feed into the TCA cycle end up as carbon dioxide. There's no viable path to regenerate any of the TCA cycle intermediates. Okay, that now moves us on to the new topic for this week, and that's the electron transport chain. Today, we're going to focus on the machinery, thinking about how this whole process happens, right? So how we take electrons stored in our 2 electron carriers, NADH and FADH2, and how we use those electrons to drive the synthesis of ATP. Okay, so today we're going to think about the machinery, and then on Thursday we're going to think about the energy aspect of it, right? So, as I hope you all have learned at some point in your biology background, right, the idea about how ATP is made is the chemiosmotic theory. Okay, and this seems like a busy slide, and it kind of is, but the idea is fairly simple. And the idea is that we produce ATP using this enzyme ATP synthase, and the energy used to drive the synthesis of ATP comes from this proton gradient across this impermeable membrane. Okay, so the inner membrane of the mitochondria, or the plasma membrane in bacteria, or the thylakoid membrane in chloroplasts. Like, the idea is that these are impermeable membranes to protons, okay? And there's a gradient where there's a high concentration of protons on the outside of the membrane, a low concentration on the inside. So there's a concentration gradient and a charge gradient that is stored energy, right? That gradient is stored energy. So as we pump protons from the outside to the inside, we can harness that energy and use that energy to drive the synthesis of ATP, okay? So we know it's this chemical energy stored as this proton gradient that is what drives the synthesis of ATP. And that proton gradient is created through passing electrons from one of our reduced cofactors, one of our electron carriers, all the way to molecular oxygen. So in aerobic organisms, oxygen is the terminal electron acceptor. We oxidize carbon all the way to carbon dioxide. We store those electrons either as NADH or FADH2, and then we pass them through a series of proteins that are embedded in the membrane of either the mitochondria or the plasma membrane or the thylakoid membrane, depending upon what organism we're talking about. And the movement of these electrons is down an energy gradient. So as we move electrons from either NADH or FADH2 to oxygen, we are producing energy. It's spontaneous to move these electrons to oxygen. That energy that was stored in NADH or FADH2 is released, and that's what drives the pumping of protons against this energy gradient, against this chemical gradient. Okay? And it's that chemical gradient then that is used to power the synthesis of ATP. This is the chemi-osmotic model. Okay? So having a general understanding of this is important as we move forward. Okay, so most of what we're going to talk about for today is thinking about the machinery for pumping, for passing these electrons through a bunch of different cofactors, ultimately all the way to oxygen. And then as time allows, we'll then look at ATP synthase. But we may end up finishing up with ATP synthase on Thursday. Before we get any further, let's think about the mitochondria, right? So this process of the electron transport chain and ATP synthesis happens in the mitochondria of eukaryotes, right? It can't happen in the mitochondria of prokaryotes because, of course, the prokaryote doesn't have a mitochondria. So instead, this happens in the plasma membrane. But for organisms that do have mitochondria, this happens there. Okay, and if we think about the anatomy of it, there's two membranes which creates essentially four compartments. Okay, there's the outer membrane, which is highly permeable. Lots of things can easily diffuse from the cytoplasm into the inner membrane space. The inner membrane space, as the name implies, is the space between the inner membrane and the outer membrane. And in this diagram, the inner membrane is shown as blue. And then inside of that is called the matrix. Okay? The matrix, we've already seen, is where all the TCA cycle reactions happen. That's also where one reaction of gluconeogenesis happens. Okay? The outer membrane is really permeable. Lots of things can easily pass through it. The inner membrane is highly impermeable. Okay? Very few things can pass from the inner membrane space into the matrix. The things that can pass are facilitated by carrier proteins. Okay? Things like ATP, ADP, pyruvate. Right? These are all things that we know we need to transport across this membrane. So there are specific carrier proteins that do so. Okay? All of the machinery for the mitochondrial electron transport chain and oxidative phosphorylation are housed in this inner membrane. So the idea is that as we transport electrons through these carrier proteins, we pump protons from the matrix out to the inner membrane space. We are moving protons outside of the matrix and building up that chemical gradient across the inner membrane. Okay? So here's kind of a big overview of the electron transport process. There are four important proteins. Only three of them are important for any given electron origin. Okay? These things are cleverly named complex 1, complex 2, complex 3, and complex 4. Okay, there are more technical chemical names. Complex 1 is NADH coenzyme Q oxidoreductase. And as the name implies, it's responsible for taking electrons from NADH and giving them to coenzyme Q. Okay, we'll look at coenzyme Q here shortly. Complex 2 is, we've actually seen part of this before. Complex 2 composes of a succinate dehydrogenase and a coenzyme Q oxidoreductase. So succinate hydrogenase, dehydrogenase, is part of the TCA cycle, right? When we think about this in the context of the mitochondrial electron transport chain, we call it succinate Q reductase. So we ultimately take electrons from succinate, creating fumarate. We know that happens in the TCA cycle. Those electrons are ultimately given to coenzyme Q. So either complex 1 or complex 2 result in a reduced form of this molecule, coenzyme Q. Coenzyme Q is the carrier molecule that takes electrons from one of these two complexes and moves it to complex 3, which is sometimes called the BC1 complex. Here, we're going to call it, so this is Q cytochrome C oxidoreductase. So it oxidizes coenzyme Q, and it reduces this protein called cytochrome C. Okay? Cytochrome C is a water-soluble protein present in the inner membrane space that then carries electrons from complex 3 over to complex 4, and complex 4 is named cytochrome C oxidase. It oxidizes cytochrome C and ultimately catalyzes the transfer of electrons onto molecular oxygen, producing water. Okay? So the electron transport chain can start at either complex 1 or complex 2, depending upon what is carrying the electrons. Any NADH that we produce in the glycolysis or the TCA cycle ultimately come in and feed electrons into complex 1. Anytime we are using succinate to reduce FAD, that's complex 2. And the one thing I want you to really note here and think about is the number of protons that get pumped in response to each of these electron sources. Complex 1, starting with NADH, results in pumping 4 protons by complex 1. Complex 2, however, note, does not pump protons, and that's important. Okay? So if the electron origin is NADH, we start at complex 1 and we get these extra 4 protons that are pumped. If the origin of electrons is the FADH2 produced by succinate dehydrogenase, then we lose out on those 4 protons. And remember, it's pumping protons that ultimately yield ATP. Okay, that becomes really relevant. Complex 3 ultimately pumps 4 protons, and complex 4 pumps 2 more. So overall, if we start at NADH, we pump 10 protons. If we start at FADH2, we pump 6 protons. Okay, that's an important thing to keep in mind moving forward. Each of these enzymes are responsible for moving electrons from one point to another, right? They are basically electron relay proteins. We take electrons from NADH, pass them through a series of prosthetic groups, redox cofactors, that are strongly bound, often covalently bound, into in these enzymes. We pass them through these prosthetic groups and ultimately give them to an electron acceptor. Complex 1 and 2, that electron acceptor is coenzyme Q. Complex 3, the electron acceptor is cytochrome C. And complex 4, the electron acceptor is oxygen. Okay? These prosthetic groups, these electron transport cofactors, fall into a number of categories. We have a couple flavins. We have in complex 2, we have FADH2. We also have an iron sulfur cluster. In complex 1, we have a different type of flavin. This is a flavin mononucleotide. The only difference is it doesn't have the AMP coupled to flavin. And we'll look at this in a second. Okay, we also have iron sulfur clusters in complex 1. So complex 1 and complex 2 both consist of flavin electron carriers and iron sulfur clusters. Complex 3, we have several heme groups. We have a heme BH, a heme BL, and a heme C1. We know hemes are porphyrins with an iron in the middle. And then we also have an iron sulfur cluster. And then finally, complex 4, we have a couple hemes and a couple copper sites. Okay? The theme here is that all of these are either organic carriers of electrons like flavin, right? We know we can reduce flavins, or they are cofactors that contain metals, copper or iron. Copper and iron are two redox active metals in biological systems, right? Those are really the only two that are easily oxidized and reduced within the constraints of the cell. All right, thinking about this in the context of energy, okay? So on the y-axis, this is the reduction potential or the free energy of electron transport relative to oxygen. Okay, oxygen is at the bottom. So the idea here is passing electrons from NADH to molecular oxygen releases energy. It's a downhill slope. Same thing starting with FADH2. Okay, NADH is a little bit higher, so the difference in energy between NADH and oxygen is a little bit more. So we produce a little bit more energy when electrons start at NADH relative to FADH2. When we start at NADH, we pass the electrons through complex 1, ultimately giving them to this little purple blob that represents coenzyme Q, or also called ubiquinone. If we start at FADH2, this is our complex 2. We pass them through our complex 2, ultimately giving them to this purple blob. This is our coenzyme Q. Coenzyme Q carries it to complex 3, which moves electrons ultimately to cytochrome C, which moves electrons ultimately to complex 4, which ultimately reduces oxygen to make water. Okay? The important thing to take home here is that each step is energetically downhill. Each step produces energy. Each of these steps produce energy which can be harnessed to pump protons against a chemical gradient. All right, let's look at the structure of coenzyme Q. Okay, coenzyme Q, ubiquinone, you'll hear me say it interchangeably. The idea is that just like NADH, just like FADH2, it can exist in an oxidized form or a reduced form in which it has accepted 2 electrons. Okay, like flavin, it's able to go through a single electron intermediate that's radical. So the 3 forms of coenzyme Q that are relevant. The fully oxidized form, we have 2 ketones. Okay, this is called ubiquinone. So both of these oxygens at the top and bottom we noticed are in the ketone form. This is the fully oxidized form. On the right here, this is the fully reduced form. This is QH2. Both ketones have been reduced to alcohols. This corresponds to a 2 electron reduction. Okay, it's important to note here that this molecule is absolutely capable of undergoing a single electron reduction. Note here, right, that in this intermediate semiquinone, one of the oxygens is a radical and the other one is either an alcohol or a deprotonated alcohol. Okay, this actually becomes really relevant. Okay, we also know that releasing radicals into our aqueous environment is dangerous, right? So we are never going to release the single electron reduced coenzyme Q. It'll never be released, okay? A couple notable things. One is this molecule is essentially all hydrophobic. So it's always embedded in the inner membrane. It never really leaves the inner membrane, okay? But also it's typically trapped in an active site of an enzyme, okay? So it never exits the hydrophobic part of the membrane. That's the really important part. So even if we do have this single electron reduced form of it, our semiquinone, we never really release it. So it's safe. The other type of prosthetic group we talked about that we haven't seen before are iron sulfur clusters. And in Dr. Hurlbert's class, he may have shown these to you and kind of explained their role. What I want to point out is that there's lots of different types of them. Okay, they come in many different forms. It could be as simple as a single iron atom that is anchored in place by bonds with 4 cysteines. Okay, this is considered an iron sulfur cluster. It is simply an iron site bound to the enzyme. More typical iron sulfur clusters are the 2 iron 2 sulfur or 4 iron 4 sulfur. The idea here for the 2 iron 2 sulfur, right, is there's 2 irons with 2 sulfurs bridging, and then each iron is covalently linked to 2 cysteines. So this iron sulfur cluster consists of 2 iron atoms, 2 sulfur atoms, anchored in place through coordination with cysteine side chains. The same thing goes with a 4 iron 4 sulfur, except this time there are 4 irons and 4 sulfurs. Okay. Functionally, they all serve the same role. Functionally, they're all able to accept 1 electron and then donate 1 electron. So they are single electron carriers. Okay. Energetically, the difference is that the reduction potential of the 2 iron 2 sulfur cluster is different than the average potential of a 4 iron 4 sulfur. It is more favorable to reduce a 4 iron 4 sulfur than it is a 2 iron 2 sulfur. And that's going to be important in the order of these things when we're thinking about our electron transport chain. Okay? The other cofactor we have seen before, but not in this form, is flavin. Right? We've seen flavin as FAD, FADH2. This is FMN. It's flavin mononucleotide instead of flavin dinucleotide. So the only difference, right? So here we're seeing flavin mononucleotide. You notice that it's a ribose attached to our flavin moiety. This is the flavin mononucleotide, as compared to FAD, which is the flavin mononucleotide linked up to a diphosphate and basically linked up to an ADP. So electronically equivalent, right? The flavin part's the only part that really matters in terms of electron transport. But it doesn't have this ADP linked up to it. That's the big difference. I guess the AMP linked up to it because it does have a phosphate here. All right, let's jump into it now. Complex 1 looks massive, right? It is. It's absolutely massive. Okay, it has 2 arms, a peripheral arm, which is situated in the matrix side of the membrane. So this is the inner part of the membrane. This is in the matrix. And the transmembrane arm, as the name suggests, is embedded in the membrane. Okay? The location of proton transport is going to be up here in the transmembrane arm, so we pump protons this way. And the direction of electron transport is going to be up through here. You can kind of see the location of our iron sulfur clusters there. Okay? The idea is that NADH docks down here at the bottom of the peripheral arm. We pass electrons stepwise through our iron sulfur clusters and all the way up to our ubiquinone site, which docks up here. Okay? The overall reaction here is we have 1 NADH comes in. Ultimately, it gives 2 electrons to coenzyme Q, resulting in an oxidized NAD+ and a reduced coenzyme Q. We also pick up 5 protons from the matrix. 4 of them get pumped across. 1 of them, along with the proton from NADH, is given to coenzyme Q for it to be in its reduced form. Okay? If you look closely, you can see the iron sulfur clusters I've already pointed out. And you'll note here that they're you can kind of even pick out the steps, right? You can see how it's going from position 1 to position 2, 3, 4. You can really see that path going there. Okay, if we looked at the structure, you'd see that there are 2 kinds of them in this path. There's a 2 iron 2 sulfur, there's a 4 iron 4 sulfur. And if you look even closer, what you would see is that the 2 iron 2 sulfur is at the very beginning, and the rest of them are 4 iron 4 sulfur. And again, remember, the reason for that is that the reduction potential of our 4 iron 4 sulfur tend to be more positive than the 2 iron 2 sulfur, meaning it's more favorable to move electrons from a 2 iron 2 sulfur onto a 4 iron 4 sulfur. Okay, and the way that this electron transport works is through a process called tunneling. Those of y'all in PChem now, are you talking about tunneling yet? Or have you talked about tunneling? Okay, so you're familiar with the idea. Some of you are. The general idea is that, right, electrons aren't particles. They're just not particles. Like they are energy blobs. Okay, so the idea is that these electrons are able to amorphously move from one position to another. Okay, so we start off at flavin. So the electrons are first given to flavin mononucleotide, and then they pass through each of these iron sulfur clusters, which are spaced typically 14 angstroms or below, like in that range. Okay, so the numbers here are, so the 14.2 that I'm showing between N3 and N1b, these are center to center distances, and the 11.0 is edge to edge distance, right? So all of these edge to edge distances are within the 14 angstrom requirement for tunneling. Okay, so you can kind of imagine how it's pretty easy to take an electron, move it from flavin onto N3, then to N1b, all the way down to N2. Okay, you note two other ones that I'm pointing out here. N7 is positioned too far away from N4 for it to be important for this tunneling. This one is probably important for tuning electron potentials of some of the other iron sulfur clusters. Okay, it's not important in the actual electron transport pathway. The other one here, N1A, this actually might be important. Okay, and the reason is that we know NADH is a 2-electron carrier, right? We consistently talk about how we take a hydride, and a hydride takes 2 electrons, and it's those 2 electrons that reduce NADH. Come back. Show up. I don't know why it does that. So with our flavin, it's also a 2-electron carrier, right? We take 2 electrons from NADH, give them both to FMN to make flavin mononucleotide in this reduced form. But I mentioned that iron sulfur clusters are single electron carriers, which means we can't give 2 electrons at a time to our iron sulfur clusters. We have to pass them one at a time. We know that flavin has the ability to form electron reduced radical. Imagine how the flavin might pass 1 electron at a time, create a radical, then pass the next one. Alternatively, it's possible that this flavin passes the electron to the other iron sulfur cluster, and then it passes it back through the iron sulfur chain. Okay. This might work, just I don't know what's going on. Okay, so the flavin has 2 electrons. We know that the flavin is able to be a stable radical, right? There can be a single electron intermediate. So the idea is we can pass 1 electron from the flavin onto N3 and down this chain. The other one can either be stored as a radical on FMN, or perhaps it's also given to N1, which can then store it temporarily, give it back to flavin, and then push it back through the chain. So the idea is because the flavin is able to create a stable radical, this is a great transition from 2 electron carriers to 1 electron carriers. Okay? We pass the electrons all the way through the iron sulfur clusters one at a time, and then we give these electrons to coenzyme Q one at a time. We start with the oxidized form, we create the semiquinone, the single electron reduced form, and then 1 more electron creates the fully reduced form. Okay, and note here on the energy diagram that each of these steps is downhill. So moving electrons from NADH to flavin is a little bit favorable. Moving it down the iron sulfur cluster chain is favorable each step of the way, ultimately down to coenzyme Q. This is a spontaneous process that ultimately leads to the production of proton pumping. Okay, so the way that the proton pumping works here, right, is that we are harnessing this energy of electron transport, ultimately reducing coenzyme Q. And the way proton pumping works, right, we need a pathway to move protons from the matrix to the inner membrane space. The way this works in complex 1 is that as we give electrons to coenzyme Q, right? That negative density is sensed by a chain of glutamic acids and either lysine or histidine, right? Shown as red or blue dots here. The idea is that the new negative charge on coenzyme Q is sensed by this series of amino acid side chains. And as new negative charge shows up, that impacts the pKa of each of these in the chain, which then demands them to be protonated, right? So negative charge on coenzyme Q requires this aspartic acid to get protonated. It pulls a proton from the matrix, and then protonation of that aspartic acid or glutamic acid then causes this glutamic acid to get deprotonated. It then moves its proton out. This glutamic acid being deprotonated is then communicated to this series, which results in a proton coming in, which then is sensed by this series, which results in a proton going out. The general idea is changing protonation states, changing charge states all the way through this chain impacts the pKa of individual side chains in a break, and you can work through the first question in your activity, and I will try to figure out what in the hell is happening with this thing.